These tree-hugging crabs may look small and cute and look a little goofy as they scurry along the trunks of the mangrove trees. They are actually very important keystone animals of the
Mangrove tree crabs belong to kingdom Animalia, phylum Arthropoda, class Melicostroca, order Decapoda, family Sesarmidae, and genus species Aratus pisoni. Aratus pisoni, or mangrove tree crabs, are native to tropical and semi-tropical regions, such as South America, Florida, the Caribbean, and so on. Within these environments, they live on mangrove trees, especially the red mangrove. They're omnivores, with the majority of their diet consisting of leaves from the trees they live on, but they are also opportunistic predators of small invertebrates such as polychaetes and nematodes. Their predators include birds, mammals, and larger crabs. Sometimes, if they escape by jumping into water, they will also fall prey to fish. Their relative size and onset of maturity differs significantly between the ecologically different locations they're found, and many studies have confirmed the broad adaptability of these species, typically associating this with the differing structural development of mangroves.
These crabs live in mangrove forests, which are a unique type of wetland ecosystem that inhabit the edge of land and sea, where the trees are rooted and thrive in salty seawaters. They can be found on the coast, around tropical and subtropical areas near the equator. The map shows where most mangroves can be found, highlighted in green. They provide habitat for thousands of species at all levels of marine and forest food webs. Species range from monkeys and tigers to snails, sponges, jellyfishes, and the mangrove crab we'll be looking at. They are considered a keystone ecosystem working in alongside with seagrass beds and coral reefs. The mangrove trees trap sediment and pollutants that could otherwise flow out to the sea, and in return, the reefs protect the mangroves from the ocean waves. The ecosystem provides an ideal breeding ground for many fishes and shellfishes, including crabs. Their complex root system also cleans water by filtering nitrates and phosphates that rivers carry to the sea while keeping the salty seawaters from infiltrating on the inland waterways. The research paper we are looking at hypothesized that the contrasting environmental conditions of different mangrove ecosystems influence the morphological differences within the Aratus pisoni population. This paper looked at 5 different mangrove forests located along the northwest coast of Venezuela. The forests varied in degrees of structural development and salt levels in the water. They collected adult crabs during the rainy seasons, only selecting those with complete chelipeds and no missing limbs. The research took 10 different body measurements on each crab. This includes looking at the height of the body, the length and width of the carapace and abdomen, and cheliped length and height. Using the statistical program PermaNova, the different body measurement values were compared with each other to determine if there were any significant differences between them.
As for the results, based on statistical tests done, they found that there was a significant difference between the measurements of different mangrove sites and sexes. This difference indicates that both environmental and sexual dimorphism influences mangrove tree crabs' morphology. 5 different male groups and 4 different female groups were identified among the different mangrove sites. The crabs in dwarf mangroves, which are small mangroves that are less than 5 feet high with higher salt concentrations than normal seawater, were found to have a greater variability in chelipid morphology. The greater variability in chelipid morphology indicates that there's possible adaptations to different chemical properties as salt levels were higher in these areas. The figure shown is a morphology figure with A representing length and minimum maximum width of carapace. B stands for the height of the body. C is the female abdomen length and width. D is the male abdomen length and width. And then E is a cheliped length and height.
As shown before, the findings support the existence of sexual dimorphism due to environmental factors. Male and female body size decreases as both the mangrove's structural development diminishes and the soil salinity increases. In hypersaline mangroves, there seems to exist a compromise between growing or reproducing. There are different allocations in energy, such as osmoregulation, respiration, or temperature control, and this may change depending on the salinity of the environment. The presence of females with more robust chelae is related to an increase in the difficulty of manipulation, such as scraping, cutting, eating, of the stronger, leathery leaves seen on the mangroves, which provides a better tolerance to environmental conditions. These changes are interpreted as an important adapted strategy to changing or stressful environment, as has been observed in other marine crustaceans.
Mangroves are an important habitat and a keystone species that house many species besides Aratus pisoni. So, since the environmental conditions can impact the morphology of this one species, there are many others that could potentially be affected as well. This could create significant changes and imbalances in the mangrove ecosystem, which is a stable environment in many warmer and tropical regions around the world. Negative impacts on mangroves can also lead to an increase of coastal erosion and larger amounts of storm surges. Morphological differences of the mangrove tree crabs themselves could potentially impact predator behavior. Predators of Aratus pisoni, including the white ibis, may select for different prey if the morphology of these crabs continues to change.